Hello everybody, in this video lecture we will cover chapter 21, Immune System, Innate and Adaptive Body Responses. So let's go ahead and begin. Okay, so immunity is resistance to disease, and immune system has two intrinsic systems. So it's kind of like divided between two systems, but they of course interconnected, and it's innate or non-specific defense system and adaptive or specific defense system. So here on this diagram, you can see how immune system is divided into innate or non-specific and adaptive or specific. Another name is natural immunity, acquired immunity. And it's also divided into lines of defense. So we have 3 lines of defense. And the first line includes skin, mucous membrane, secretion, and normal flora. It can be normal flora of your skin or flora of your intestine. The second line of defense include immune cells. It says innate immune cells. So it's pretty much all the cells except B and T cells, except of these lymphocytes. Everything else, all other cells can be part of the second line of defense. Well, of course, immune cells, not every single cell, right? But all other immune cells, rather than lymphocytes, are part of the second line of defense. Inflammation, complement, antimicrobial substances. And third line of defense include B cells and T cells. And we will talk about it in more details, right? So innate defense system has 2 lines of defense, right? That's what it says. Innate has 2 lines of defense. First line of defense is external body membranes, skin and mucous membrane. And I'm reminding you that mucous membrane cover all your open body cavities, right? Second line of defense is antimicrobial proteins, phagocytes, and other cells, inflammation, and fever. And the second line of defense inhibit spread of invaders, and inflammation is the most important mechanism in the second line of defense. An adaptive defense system is a third line of defense that attacks particular foreign substances, takes longer to react than the innate system, and they are deeply intertwined, right? Both innate and adaptive. So over here, you can also see another diagram that pretty much tell us similar, right, information, gives us similar information. So innate, adaptive, and this is first line of defense, second line of defense. And both of these, humoral and cellular immunity, is the third line of defense, right? And you can see again here, skin, mucous membrane, and then second line, phagocytes, natural killer cells, inflammation, fever, antimicrobial proteins. And adaptive defense is divided into humoral immunity, and humoral means this immunity happens inside fluids of your body, right? It can be tissue fluids, right, for example. Cellular immunity happens kind of within the cells. So when cells are affected, right, we do have some mechanism to destroy those infected cells and as a result destroy those viruses or bacteria that are intracellular parasites. Okay, so in the second line of defense, we have inflammatory response, right? So it's over here, inflammation. So it's the most important out of, this, it's, well, of course, everything is important, but it's a really important process. That's why we're looking at it in more detail. So what is inflammation? What is that inflammatory response? So first of all, it's triggered whenever body tissues are injured or infected. And it prevents the spread of damaging agents. It disposes of cell debris and pathogen and sets the stage for repair. Now, those are cardinal signs of acute inflammation, and it include redness, heat, swelling, pain, and sometimes impairment on function. Now, what happened when your tissue became damaged? And it can be, you know, it can be different type of damage, right? You can cut yourself, that's a damage. Or bacteria can, you know, live in your throat, start reproducing and secreting different toxins that, that also damage your cells. So either you cut yourself or you have bacterial infection, you have viral infection, your cells are damaged because your whole body is made of cells. So when your cells are damaged, right, so you have your cell and there is a damage to your cells, right? So either some physical damage, right, like you're cutting it, or some, you know, microorganism affecting it. But those injured cells, right, so when they injured, they start secreting chemicals, right? Like saying, help, right, we are in trouble. Now, this is called inflammatory chemicals. There are different types of inflammatory chemicals. Some of them are prostaglandins, right? It can be histamine, right? Also inflammatory chemical. But when you damage cells, secrete chemicals, and also white blood cells that are attracted to the site of injury, they also secrete chemicals. Now, these inflammatory chemicals, they cause blood vessels to dilate, right? So it's what it says, capillary widening. So capillaries became wider, so dilation of your capillaries. And it's increased blood flow. So when you have injury to the cells, you want more blood flowing to this area because your blood can bring oxygen, it can bring clotting factors, repairing factors, it can bring nutrients, right? So you want lots of blood flowing to the damaged area. So capillaries became wider and increased blood flow. And what happens if blood flow is increased? It gives you heat and redness, right? So that's redness and heat because of the dilation of local capillaries at the site of injury, redness and heat, right? And plus those chemicals, they make capillaries more permeable. And when capillaries became more permeable, what happened? Fluid release into tissue, and this causes swelling. And also this chemical activates pain receptor, right? And over here it says systemic response, fever and proliferation of leukocytes cause pain. That's right, fever, proliferation of leukocytes, but also the chemicals itself will activate pain receptor and it give you pain, right? It also has here tenderness, but you remember this four major: redness, heat, swelling, pain, and sometimes impairment of function, right? So, and it's all caused by what? Because of this inflammatory chemicals are released either by white blood cells or damaged cells or both, right? And it causes dilation of capillaries, right? Heat, redness, capillaries became more permeable, that's a swelling, and this chemical activates pain receptor. And plus, of course, you have a systemic response that also activates pain receptor. So that's a pain. So what are those chemicals, right? Chemicals that are released either by your damaged cells or some cells of the immune system. Inflammatory mediators, histamine. Histamine, you probably already know that histamine causes the dilation of blood vessels. So histamine released from mast cells, blood proteins such as, right, that your blood, when capillaries dilate, you have blood proteins that are going to be delivered to the site of the injury, and it will include like kinins, prostaglandins. I don't know, in a 50A, you should cover how prostaglandins are synthesized, right? They synthesize from phospholipids of cell membrane, right? So kinins, prostaglandins, some interleukins, or leukotrienes, complement, right? Those are all inflammatory chemicals. And they are released by injured tissue, phagocytes, lymphocytes, basophil, mast cells. Now, basophil also release histamine. Now, inflammatory chemicals, and I already told you that, right? Because I told you here what those inflammatory chemicals do, but here you can read about it. They cause dilation of arterioles, resulting in hyperemia, increased permeability of local capillaries and edema. It's a leakage of exudate. Yeah, if I told you dilation of capillaries, I apologize, because capillaries, they don't really have smooth muscles, right? So they cannot dilate much, right? So they cannot dilate. So arterioles, small arterioles still have smooth muscles, so they will dilate. Capillaries became more permeable. So I apologize for that. Now, this exudate contains proteins, clotting factors, antibodies, and function of this surge of exudate is to move foreign materials into lymphatic vessels and deliver clotting proteins to form a scaffold for repair to isolate the area, right? So on this diagram, you guys can now see in a full detail when your tissue is injured. And on this picture, it shows you like some mechanical injury. Somebody just cut himself, right? But it doesn't necessarily need to be a cut. It can be bacterial infection as well, as I told you, viral infection. It can be some fungus, right? It can be protozoa, anything that is damaging your cells, right? So when you have tissue injury, you have release of these chemical mediators or inflammatory chemicals, including histamine, complement, kinins, prostaglandins, and also releasing leukocytosis-inducing factor. Right, so let's look at this part first. So we have release of these chemicals into an area of injury, right? And what's happened? It will vasodilate arterioles, right? Again, arterioles, because they do have smooth muscles, so they can dilate and they can constrict. It will increase permeability of capillaries, and it also will attract neutrophils, monocytes, and lymphocytes to the area, what we call chemotaxis, right? So this is all the cells of the of innate immune response, with exception of lymphocytes, right? So they will also be attracted through chemotaxis to the site of injury. Now, when we have dilation, we have hyperemia, so increased blood flow to the area. This is what hyperemia is. Hyper means a lot, emia means blood, lots of blood, and it will give you heat and redness. When we increase the permeability of capillaries and we have leak of exudate, then this bring rich proteins, fluid rich with the proteins, and it will cause swelling because we have this leakage and pain. Right now, this exudate supposed to be picked up by your lymphatic vessels, right, and remove possibly all, you know, this either some pathogens or maybe some toxins, right, from the site of the injury. So here's our cardinal signs: heat, redness, pain, swelling. And because we have pain and swelling, we have possibly temporary limitation of function. Here it says joint movement, but can involve other function as well. This locally increased temperature, increased metabolic rate of cells, and it all leads to the healing. Also, clotting proteins prevent injury from spreading through the tissue, and temporary fibrin patch forms, right, scaffolding for repair, so it also lead to healing process. And over here, when we have this leukocytosis inducing factors, we increase the number of leukocytes in a bloodstream, and they can migrate to the area through diapetesis, right? And diapetesis is like movement of amoeba, if you remember from biology class, and they will phagocyte, so it will be phagocytosis of pathogens and dead tissue cells by neutrophils and macrophages, and it will clean the area. So it all leads to healing process. So you can see over here why inflammation is such an important process, right? Because it activates so many responses over here that will cause healing of the injury. But you also can see that it's not specific, right? It doesn't have any memory. Next time when you cut yourself, you start all over again, right? You don't have a special cells involved here that respond to a specific type of injury, right? That's why it's innate, right? That's why it's non-specific. So this is our second line of defense, right? Inflammation. Another part of second line of defense or non-specific defense is complement. You know, like nice word over here, but what is complement? Complement is actually about 20 different blood proteins that circulate in your bloodstream in an inactive form. Most of them are synthesized by your liver. So your liver synthesizes proteins and they just circulating in your blood, right? And they have names. So they include C1, C2, C3, C4, C9 factors. So we call it this way. Then factor B, D, P, regulatory proteins. And now when you have infection, then this complement will be activated. And when complement is activated, when those proteins are activated, they actually form a specific complex. So they kind of like, you know, you have proteins and they form this special complex, right? And it can destroy bacteria. And we will look at it in more details in a minute, right? So when this protein, when this complement is activated, this active complement can destroy foreign substances. And it also enhances both non-specific and specific defenses. Oh, here, by the way. Okay. So that's our complement activation. We don't need to go too far in our lecture. So here, what you see, okay, so this is bacterium, right? Now, this blue part is antibody. So your, your body already responded by producing antibodies toward this bacterium, and this antibody activates our first protein in this complement system, right, C1. And then it's caused a chain of activation, sequential binding and activation as a factors until the whole complex is formed called MHC or membrane attack complex. So this complex, this MHC, gonna make a hole inside the bacterium, right? So it's really, it's kind of it get incorporated and it's make like a little tunnel. So that would be this MHC. So it's gonna be the whole complex that make like a little channel. And what happened, because we have like inside the bacterium is hypertonic solution, right? So the water will just move in through this opening, and it will cause lysis because of huge, massive influx of water, right? So that's a complement. So again, what is complement? Those are proteins circulating in your blood, and if you have a bacterium, right, and so that complement can be activated, forming MHC, membrane attack complex. They're gonna do holes inside bacterial membrane, and it will cause death of bacteria. Now, fever is also second line of defense. It's a systemic response to invading microorganism because leukocytes and macrophages, when they exposed to foreign substances, they secrete pyrogens. So those are chemicals that gonna reset your thermostat upwards and your body will have a fever. Right, you know that we already covered homeostasis for body temperature, how your body has, regulates your internal body temperature, keeps it in a limited, within specific limit, right? So, and this sensor for your body temperature is in hypothalamus, right? But when your body has an infection, those pyrogens, they restart the thermostat, and now your body thinks the higher temperature, that's your normal temperature, right? So your temperature rises, and that's why you have fever. Now, fever, when it's high fever, it's dangerous because it can be nature enzymes. But moderate fever is beneficial for you. And what fever does, when you have moderate fever, it causes liver and spleen to sequester iron and zinc. So you have like, you don't release this iron and zinc. And this is what needed by microorganisms for growth and replication. And plus, fever increases metabolic rate, which speeds up repair. So, right, benefits of moderate fever include the low amount of iron and zinc available for microorganisms, and also increases metabolic rate for repair of your tissues. Okay, so what we talked about over here, I will go back, and this was our second line of defense. We didn't cover much your skin and mucous membrane because in Bio 50A, you guys study the structure of skin and mucous membrane, right? And we talk about here about inflammation, complement, antimicrobial proteins, and fever. Phagocytes and natural killer cells. Phagocytes are important for both innate and adaptive response. And natural killer cells, we didn't cover here, but they destroyed let's say they do destroy your body cells that are infected or damaged, but they are not specific. They pretty much don't develop any memory, right? And they just destroy everything that does not belong to your body, that is non-self. Right, so that was our innate defense. So now we're moving to adaptive defense. And of course, if you take microbiology, you guys will cover everything in way more details. So take it just like introduction to your microbiology course. Now, adaptive defense. How it is different? Because it is specific. It is systemic. That means many body organs and systems are involved, and it has memory. And adaptive defense has two overlapping processes or arms. It's humoral or antibody-mediated immunity, and cellular or cell-mediated immunity. So remember, humoral, something happens inside the fluids of your body, and cellular related to the cells, right? So before we start talking about humoral immunity, let's talk about antigens. Antigens are substances that can mobilize the adaptive defense and provoke immune response. Most of them are large, complex molecules not normally found in the body, right? So it can be bacteria, it can be virus, right? It can be some protist, but it also can be toxins of this microorganism, right? So those are just large molecules, usually proteins, that your body recognizes as non-self, right? We have complete antigens and we have haptens. So let's see what is the difference. So complete antigens, they have important functional properties such as immunogenicity. And genicity, genesis means you generate something. So you generate immunity. So immunogenicity is ability to stimulate proliferation, and proliferation is division, right, through mitosis. proliferation of specific lymphocytes and antibodies. Antibodies, of course, do not use mitosis, only cells can do, right? But lymphocytes will divide, and special type of lymphocytes like plasma cells will secrete antibodies. So if it's a complete antigen, it has this ability to stimulate proliferation of lymphocytes and antibodies, and it's also have reactivity, ability to react with product of activated lymphocytes and antibodies released. So they activate lymphocytes, they react with lymphocytes. They activate lymphocytes to secrete antibodies, and they react with antibodies. An example would be large molecules like foreign proteins, right? Usually proteins, very often like it says here, polysaccharides, lipids, nucleic acid, but the most powerful complete antigens are proteins. And haptens are small molecules like peptides, nucleotides, hormones, right? If here we have like larger molecules, here we have smaller molecules, and they're not immunogenic by themselves, but they are immunogenic when attached to body proteins. And this is, you know, what you find, for example, in pollen, right? So when people have allergies on the pollen, that would be an example of haptens. So haptens causes the immune system to mount a harmful attack to something that is not really dangerous, like poison ivy, animal dander, detergents, cosmetics, right? Something that you call like, oh, I have allergy to something. Like haptens by its own, they do not produce immune response, but when they attach to our own body proteins, then they can induce immune response. Now, so we talk about antigens, right? So those are complete antigens, haptens, but those antigens, so for example, that's an antigen, and we can, you know, let's say it's a bacterium, just to make it easy. Like maybe this is a bacterium, right? And we call it antigen. Why we call it antigen? Because it has this immunogenicity and reactivity. So it will stimulate your lymphocytes, right? And it will react, right? So that's with lymphocytes and antibodies. So that's a bacterium. But on the surface of the bacterial cell wall, we would have antigenic determinants. So determinants are specific part of entire antigen that are immunogenic, right? So it's not like every single part of this membrane that is phospholipid membrane, not every single part of this membrane is immunogenic, but special parts are. So these parts will provoke this immune response, and we call it determinants. And antibodies, so here antibodies shown in blue, and lymphocytes receptors can bind to them. And most naturally occurring antigens like bacteria, viruses, they have numerous antigenic determinants, right? So over here you can see how they all have different shapes. Now, our own cells, right, have what we call MHC, or major histocompatibility complex, MHC proteins. Those are self-antigens on the surface of the cells. And this is how your own immune system recognizes your cells. The MHC proteins, they're antigenic to others, right? So when we have transfusion or tissue drafts, right, that's why we have rejection of somebody's tissue, because their immune system doesn't recognize MHC protein as self-antigens. All right, those are unique to an individual. Okay, so we cover antigens, right? We covered antigen determinants, and we covered even our self-antigen very briefly. Now let's look at the cells of adaptive immune system. So if you go back over here, and as I told you, that in the second line of defense, we have innate immune cells. And the third line, we have lymphocytes, right? So all other immune cells are not part, well, they do play a role in a, adaptive immunity, right? But they're not considered to be cells of the specific response. Lymphocytes are the cells for adaptive immunity, right? So, okay, where's our lymphocytes? Okay, so two types of lymphocytes. B lymphocytes, or we call them B cells, for humoral immunity, and T lymphocytes, or T cells, for cell-mediated immunity. And another cells that part of adaptive immune system are antigen-presenting cells that are not specific. Those are specific, right? They will be activated by specific antigens. Antigen-presenting cells are actually phagocytes, right? Or dendritic cells, or macrophages, right? They do not respond to specific antigens. So they are part of both specific and non-specific immune response. But they play essential role in both of these immunities, right? Now, we will talk about these antigen-presenting cells. Everything will make sense in a minute. But let's begin with lymphocytes. Lymphocytes originate in red bone marrow, the same as any other blood cells. Now, after they originate in red bone marrow, I'm sorry, originate in the red bone marrow as any other blood cells. But after that, B cells, they mature in the red bone marrow, and T cells mature in the thymus. And I'm reminding you, thymus is the organ that located superior to the heart, right, on the top of the heart. And when they mature, what does it mean they mature? Well, that means they became immunocompetent. So first they, they are not mature in the red bone marrow, then they move to the thymus or stay in the red bone marrow, and they became immunocompetent. And that means that they are able to recognize and bind to a specific antigen. So they learned, right? Well, they don't learn, but they are they selected. Only the ones that can bind to specific antigen, they selected, and they can then divide. So they became immunocompetent, and they became self-tolerant. Self-tolerance means they are unresponsive to self-antigen. So you need to bind to specific antigens, and you don't have, and you cannot or must not bind to self-antigens, right? So immunocompetence and self-tolerance. When they became immunocompetent and self-tolerant, right, and they still stay in the thymus and bone marrow, right, we call them naive. Naive means they were never exposed to pathogen yet. And those naive B and T cells, they move from bone marrow and from thymus to lymph nodes, spleen, and other lymphoid organs, and they stay there until they meet with their antigen, right? That makes sense. So maybe this diagram makes it more clear. So here we have immature lymphocytes in the red bone marrow. Then they migrating into thymus, so they will be T cells, stay in the bone marrow, B cells. And in thymus and bone marrow, they became immunocompetent, right? So they can bind to the antigen, and they do not bind to self-antigen, but they're still naive. So they didn't, so and again, naive means they are unexposed to any bacteria, any virus, any other pathogen yet. And then they move to lymph nodes, right? And other lymphoid tissue like spleen. And they, what it says, they see it over there. And this is where they encounter their antigen. So, so pretty much to make, so for example, right, so you have your cells, right? So here's your cells and they, right, so now we have some pathogen, right, maybe some bacteria entering. And now what will happen on the site of injury, you will have like cell neutrophils, right? So here's the neutrophils, and neutrophils are white blood cells. So they are phagocytic. So they're gonna engulf this, right? So they're gonna pretty much eat it, right? Okay, so here's inside, we have this bacteria inside that this neutrophil gonna eat, and it's gonna release pieces of it, right? So here's the pieces of this bacteria, and this is called antigen. And those antigen will be picked up by your lymphatic vessels, right? So here we have, it's a dead pathogen, but we have lots of them here. You know, we have so many bacteria, right? Because they reproduce very, very quickly. But for example, you have some of them that was destroyed by neutrophils, right? So they destroyed by neutrophils. So neutrophils pretty much digest them, right? And they release pieces of this pathogen into your tissue. So here we have pieces of pathogen with the fluid. Now this fluid and this antigen is picked up by lymphatic vessels, and you know lymphatic vessels move to the lymph node. And what is inside this lymph nodes? This is our naive B and T cells already over there. And this is where they encounter their antigen. Right? That makes sense? So now antigen activated immunocompetent lymphocytes circulate. Now, when they activated by antigen, they circulate in the bloodstream and lymph and through the lymphoid organs of the body. Okay. Now, T cells mature in the thymus under negative and positive selection pressure. Positive selection selects T cells incapable of binding to self-MHC proteins, and negative selection prompts apoptosis or death of T cells that bind to self-antigen displayed by self-MHC, right? So here in the thymus, we have maturation of T cells. So you have, like shown here, like 5 different T cells. Now these T cells, they bind to your own self-antigen, so they will be destroyed by apoptosis. So that's negative selection. And the ones that do not bind to self-antigen, through the positive selection, right, they move right now from the thymus to the lymphatic organs, and this will be our positive selection. Okay. Now, antigen receptor diversity. Lymphocytes make up to 1 billion different types of antigen receptors, and they're coded by 25,000 genes. So over here, all these cells, it can be B cells, for example. So look at all these B cells, and they all have different receptors, like billion of different receptors, to make sure that any antigen enters your body, any bacterium, any pathogen, other pathogen enters your body, you would have a B cell that has specific receptor for this specific antigen, right? Out of billion different receptors, there is high chance that one receptor will feed this antigen. And when this antigen feeds to this receptor, this specific cell is activated and it goes through the clonal selection. That means this cell only will divide. Because every time when there is a bacteria of virus in your body, you don't want all B cells, all T cells, all those billions different type of cells start dividing. You want only specific B and T cells to be able to divide, right? And this specific will be the one that has receptor for that specific antigen. Now, antigen presenting cells. So I hope we have like understanding what are B cells and T cells are, right? Where they are formed, how they move to the thymus or stay in the bone marrow, where they became immunocompetent but still naive because they never expressed, they are not expressed yet to the pathogen. So those naive B and T cells, they move to lymphoid organs, and that's where they stay until they are activated by their specific antigen, and then they start circulating in your blood and your lymph, right? So that's B cells, T cells, and now antigen presenting cells. Now, antigen presenting cells, they engulf antigen. So this is macrophage, for example, or dendritic cells. So, so here we have bacterium enters your body, right? Obviously, this will promote your non-specific response. And non-specific response include migration of macrophages and neutrophils to the site of injury, right? So that's macrophage moves to the tissue that is injured, and it's what it does. It's actually ingests this foreign antigen, right? Here it has lysosomes fuses with this phagosome, and what is in lysosome? Digestive enzyme, right? So it's pretty much break it apart, right? So it's digests this bacterium. And of course, lots of this will be maybe removed from the cell, but pieces of it will be presented on MHC protein. So there's a MHC protein. Remember, that's our own proteins, right? So they can be like class 1, class 2, right? But piece of this antigen are presented on the surface of this macrophage. So this macrophage kind of now like here, there is an bacteria inside our body, and here a proof, right? Here's a piece of it, right? This is a fragment of this bacterium. And this fragment, it has specific determinants. So if I go back over here, it has specific determinants, this shape or this shape or that shape, right? So this is what macrophage presenting to your actually T cells, right? So now helper T cells has T cell receptor and other special receptors that would recognize the shape, right? I think we're gonna look over here. Okay. So let's see. It's somewhere over here, way down, but so here the T cells, right? And it has T cell receptor and some other receptors that make them helper cells like CD4, right? So all different receptors on the T cells, but T cell receptors, they bind to this fragment of antigen presenting cell. Right, so let's go back. Okay. So here we have macrophage presenting this fragment of antigen to a helper T cell. Helper T cell has this T cell receptor that need to fit in the shape of this antigen, and then it became activated and it start releasing chemicals that we call interleukin. Leukin means leukocytes and inter means between. So those chemicals that allow leukocytes or white blood cells communicate with each other. So it's called interleukin. Right, and then we will follow those interleukins and we will see what those interleukins are gonna do. So far, why we're talking about it, because we said that the cells for adaptive immunity include B cells, T cells, and antigen presenting cells, right? Now B cells, They don't need antigen-presenting cells to be activated, but T cells do. T cells need these antigen-presenting cells to be activated. Now, how these antigen-presenting cells activate T cells? This is what is shown on this diagram. Right? Now, antigen-presenting cells are dendritic cells in the connective tissue and epidermis, macrophages in connective tissue and lymphoid organs, and B cells. And it can be some atypical APCs, but those are major players, major antigen-presenting cells. Now, here we're talking about humoral immunity. Now, antigen challenge. First encounter between antigen and naive immunocompetent lymphocytes. So remember how these immunocompetent lymphocytes, but they still naive, how they sitting inside lymphoid organs. For example, inside a lymph node. So they sitting there inside a lymph node, and then if your body has some antigen, those antigen move through the lymph inside this lymph node, and we have the first encounter between lymphocytes and antigen challenge. So usually occurs in a lymph node or in the spleen. Now, if this lymphocyte is a B cell, then it will provoke humoral response, and your body will produce antibodies. Right, so let's look over here. So, so here, first of all, where are we at? We are right now inside, for example, lymph node. So it's some lymphoid organ like spleen or lymph node. So inside your lymph node, you have these B cells. They are immunocompetent, but they're still naive, right? So immunocompetent means they can bind to antigen and they do not bind to self-antigen. So they know difference between your cells and the foreign cells, right? So they sitting inside this lymph node. Now, what happened over here? Antigens entering lymph node. How antigens enter lymph node? So, for example, if you have like, so here's you, right? Oh my goodness, it's not you, it's some, you know. And this person has a sore throat, right? Sore throat, what it means? It means there is some bacteria over here, right? So bacteria. So if you look this, so here's the bacteria, right? Here's a bacteria inside this throat, and it starts damaging the cells. Of course, cells start secreting these inflammatory chemicals, right? We have inflammation, but then we have, for example, macrophages. So here's the macrophages. Through diapedesis, enters this area. It's actually gonna what? It's gonna ingest and eat this stuff. Okay, if it's macrophages, it's a little bit different. Let's say this is neutrophil. Okay, so what is this? It's a neutrophil. It's a type of leukocytes. So that's a neutrophil. So the neutrophil eats this bacteria because they're also phagocytic. They're gonna release these pieces of it. So they digest it and release pieces of it, and through the lymph nodes, right? And this is what we call antigen. Through the lymphatic vessels, it actually moves, right? So here's your lymphatic vessel. It moves inside the lymph node. And here we have B cells. Now, if B cells are activated, right, we will have humoral response. So now we have 3 different B cells over here. You have hundreds of different B cells. All of them gonna be activated? No, the only B cells that have specific receptors for this particular antigen will be activated. That's why we call it specific immunity. So this, you know, this cell and this cell, they continue resting, right? They are not gonna do anything. They're not doing anything. The one that has specific receptors, and remember, we have billion of different receptors. So the chances are some receptors match with antigen. Now they are selected. And when they are selected, right? They go through proliferation. So they start dividing, right? These activated B cells are dividing, and then they differentiate into plasma cells and memory B cells. Now, memory B cells now gonna circulate in the blood and lymph, right? And they will keep a memory of this invader. Plasma cells, they're gonna secrete antibodies, and they secrete antibodies that specifically match this antigen, right? So, so that's kind of like our primary response, and it immediately shows you that when several years later, when you have the infected by the same pathogen, so it's the same antigen, you already have these memory cells circulating in your blood, right? So you don't need, you kind of skipping this process from going to lymph node, right? Then activating these B cells, then proliferation. You already have these memory cells in your blood, right? So they very quickly start dividing and differentiating into plasma cells. You see, we have way more plasma cells, we have way more memory cells, right? So this response are very fast, right? So you fight the same infection more rapidly than you did the first time when you were just introduced to this specific antigen. So that's what the memory is. So that's our humoral response, right? Humoral immunity. So this is inside the lymph nodes, right? When they select it and they differentiate, B cells differentiate into plasma cells and memory cells, these cells, they start circulating in your blood, right? So they, blood brings it to the site of the injury, and here's you have your antibodies, right? And memory cells continue circulating. So just in case if you get this infection again, so you can respond more rapidly. Right, so primary immune response occurs on the first exposure to a specific antigen. A lag period is 3 to 6 days, and peak level of plasma antibodies are reached in 10 days, and then antibody levels declines, right? And secondary immune response occurs on re-exposure to the same antigen. Synthesized memory cells respond within hours, so they were synthesized before during your primary immune response. Then antibody level peaks in 2 to 3 days, not 6 days, and it's also at much higher levels. And antibodies bind with greater affinity, and then antibody level can remain high for weeks to months. So on this graph, you can see how they compare primary and secondary immune response, and what's shown in blue is the same pathogen, so it's the same virus, same bacteria. So when you just introduce first time to this infection, right, so you can see it's almost take a week for your body to start synthesize antibodies, right? And here's you do, so this is like a units, how many of these antibodies you have, and then their level declines, right? But when you expose a second time, you see how rapid this response is. So you just couple days, like 2 days, and you already produce way more antibodies, right? And the level stays high. Level of antibodies in the blood stays pretty high, right, for a long time. But if you introduce to a different bacterium, right, that's a different infection, you start all over. That's your primary immune response. But then if you already exposed to this, your secondary immune response will be more, more rapid, right, with more antibodies, and really you're gonna respond quickly. Okay, now here I will go just to this diagram, because everything else you can read on its own. This diagram explains these previous like 3 or 4 slides. So humoral immunity can be divided into active and passive. What active means, you actually have contact with pathogen, right? So you have contact with pathogen and you produce your own antibodies. And passive means you do not have contact with pathogen, you just giving these antibodies either naturally or artificially. Now, humoral immunity, active, passive, and both can be naturally acquired or artificially acquired. So active, naturally acquired will be when you have this infection. So you have infection, you have contact with pathogen, right? You go through all, these stages and you start synthesizing your own antibodies. Or you can be giving a vaccine of dead or attenuated pathogens. So the one that is weak, the pathogen that doesn't harm you, but you're giving pathogen and you start producing your antibodies. Passive, you are giving antibodies. You can be giving through placenta, right? Or through the milk in infants or fetus. Or you can also be have a vaccine, right? But it's not the pathogen. It's serum that already has antibodies. Gamma globulin are antibodies, right? Like now what they're doing this vaccine for COVID-19, and the ones that they have in Russia that, America, doesn't really think this is gonna do anything, but this would be this humoral immunity, passive, artificially acquired, because in the Russian vaccines that they have right now, they do not have pathogens, they just have these gamma globulins, right? So that's not, of course, that, this is better. This vaccine is way better when it gives you active immunity. But as long as I know about this Russian vaccine, because there is lots of information that, we cannot trust, but my understanding is that's gonna be this artificially acquired passive immunity through vaccination of gamma globulins. Okay, now antibodies produced by plasma cells, right? So another name are immunoglobulins or gamma globulins. Proteins secreted by plasma cells, and they're capable of binding to specifically with antigen detected by B cells. So here's the bacterial cells, and you see this green stuff, those are determinants, and antibodies will bind to their specific determinant. Right, so they match like a key and a lock. Now, very quickly, basic structure of antibodies, they T or Y shaped. And let's just look over here. So this is how they look. So they have two heavy chains and two light chains. And both heavy and light chains, they have constant region, right? So this is constant for light chains, this is constant for heavy, and it has this variable region. And this variable region, this is where it will bind to specific antigen. So it will have a specific shape that binds to specific antigen. And the constant region, just what constant region does, it says over here, it determines the class of antibodies. So it's immunoglobulin M, A, D, G, or E. It also determines how this antibody function, how it eliminates antigen, and what cells and chemicals they bind to, right? So that's a constant region. And the variable region has this specific shape that binds to specific antigen. Okay, so how antibodies actually work? So when antibody, antigen, so antibodies, they form the complex with antigen. We call it antigen, antibody, or immune complexes. And there are several mechanisms how these complexes work. Neutralization, agglutination are common, and there is precipitation and complement fixation. So here in this picture, you see a mechanism of neutralization. It's the simplest mechanism. Antibodies bind to their antigen, right? And it blocks specific sites of the viruses or bacterial toxins. And they prevent those antigen from binding to receptors or tissue cells. So for example, you have virus, right? Virus is intracellular parasite. For this virus to enter your cells, it has a specific receptor, right? It's need to bind to the receptors of your cells and move inside. So now if you have this neutralization, if you have this antibodies binds, right, then it would be difficult for this virus to penetrate your cells, and it would be easier for macrophages to eat it, right, to destroy it. So antigen antibody complex goes phagocytosis. So that's agglutination, when antibodies bind to same determinant on more than one cell. So here's one antibody, it binds to this cell and this cell, right? This one binds to this cell and that cell, and they form this cross-linked antigen antibody complexes, and they agglutinate or clump together, right? Like example would be clumping of mismatched blood cells. Precipitation, when we have soluble molecules and they cross-linked with antibodies, and they precipitate. So they became, from the soluble, they became insoluble, and they again can be phagocytized, subject to phagocytosis. And here the whole diagram, right? We discussed neutralization, agglutination, most common, precipitation, and here's a complement activation or complement fixation. So here we have bacterial cell, right? And antibodies bind to it, and they activate complement. And remember how complement form this membrane attack complex, and it's making kind of like a holes, and we have lysis of cells. And of course, all of these processes, they enhance phagocytosis and inflammation, and complement leads to cell lysis. Okay, so that was humoral immunity. That's why we talk about antibodies, right? So that's all that we cover so far, right, is humoral immunity. And now briefly about cell-mediated immunity. Humoral immunity happens in fluids of the body. Cell-mediated immunity is your defense against intracellular antigens. So two types, and the cells that are part of cell-mediated immunity are T cells. Now over here in blue, you see two different T cells. We have more than two, but those are kind of the major one, CD8 and CD4. Why we call them CD8 and CD4? Because they have special proteins on the surface of their cells, and that's the name of those proteins. Either they have CD4 protein or CD8 proteins, right, glycoproteins. And then they have TCR or T cell receptor, and TCR binds to MHC. This is our major histocompatibility complex, right? So on a cells, either it's your target cells or antigen presenting cells, right? Now, so here's the major T cells, CD4 cells, CD8 cells. Now CD4 cells, when they activated, they become helper T cells. CD8 cells, when they activated, they become cytotoxic T cells, and those destroys the foreign antigens, right? Cytotoxics, they're gonna kill your own cells that are infected. And here's the other T cells, like regulatory T cells, memory T cells. Now, okay, now this is what's happening here. Remember, we had similar picture when we talk about this humoral immunity, right? So that was humoral immunity. We were inside a lymph node, right? And then through lymphatic vessels, we have antigen, right, that is with lymph, enters the lymph node and activates B cells, and we have our humoral immunity response, right? And humoral immunity response is production of antibodies and memory B cells. Now, okay, now where we at? Well, here they begin with red bone marrow. Remember, this is where we have a formation of the T cells, and then they move to thymus. So here they are in the thymus. In the thymus, they mature, and so they develop these receptors, and if they develop CD4 receptors, they will be CD4 cells or CD8 cells, so they are in the thymus. Now this light brown, we again inside lymphoid tissue and organs. So we again inside lymph node. But now what happened in the lymph node? Now we have actually is a, well, here is dendritic cells, but it can be macrophages as well. So this is our antigen presenting cells. So those are macrophages. Remember that, I'll go back over here and I show you. Over here. Remember how we have, you know, somebody that has a sore throat. So here's a bacteria in the throat, and here macrophages migrate and they digest these bacteria, but they have a piece of this bacteria on their surface, right? This is macrophage. It can be dendritic cell as well. So now these cells, antigen presenting cells, they move through lymph inside lymph node, and here they add. So here they add, right there, right? So here just dendritic cells, but again, can be macrophages as well. Now those APC or antigen presenting cells enter lymphoid tissue and organ, for example, lymph node. And we have like lots of these T cells, CD4 cells, CD8 cells, and they're gonna activate these cells. Are they activating all of those cells? No, just specific cells that have specific receptor to this antigen. So they activate them. If they activate CD4 cells, they became helper T cells. And those helper T cells circulate in the blood plasma. If they activate a CD8 T cells, then they became what? Cytotoxic T cells that circulate in the plasma as well. And of course, some of them will be memory cells that will circulate in your blood, in your lymph, and again, waiting for a new exposure to the same pathogen. That's why this is our specific immunity, right? It has memory. Now, these cells now circulating in your blood, and cytotoxic T cells, I think it's over here. No, maybe before. So cytotoxic T cells, I don't know, I think it was somewhere. They're gonna destroy your own cells that are affected by the intracellular pathogen, and helper T cells has a very important job that we will cover in this slide, right? But so far, make sure you compare this diagram, right, with the one that we covered before, this B cells, right? So here's what happened inside the lymphoid tissue with B cells. And we start a humoral response, and this is what happened inside the lymphoid tissue with T cells, right? And that's the beginning of the cell-mediated response. Now here we compare humoral and cell-mediated. So antibodies for the humoral response, the simplest ammunition of the immune response, so that's really more simple, and they target bacteria and molecules in extracellular environment, body secretion, tissue fluid, blood, and lymph, right? That's a humoral response. So humoral response includes B cells, right? Antibodies, where B cells differentiate into plasma cells that secrete antibodies, and they target parasites that are in the fluids of the body, right? Now, T cells, as a part of cell-mediated response, they recognize and respond only to process fragments of antigen displayed on the surface of body cells. So T cells can only be activated by antigen-presenting cells, right? They're not gonna respond just free-floating antigens like B cells. So they need these antigen-presenting cells, right? And what they target? They target body cells infected by viruses or bacteria. So your own cells that already infected and you just want to really get rid of them and prevent those bacteria and viruses from reproduction. Abnormal or cancerous cells, and also cells of infused or transplanted foreign tissues. Okay, now role of helper T cells. So helper T cells, without helper T cells, there is no immune response. Actually, the helper T cells, this is what binds together humoral and cell-mediated. So let's look over here. So when macrophage, right, engulf this, let's say microorganism, right, some bacterium. Now what's gonna happen? It's gonna chop it up, digest it, right, into small fragments. And some of these fragments will be will be presented on the surface of macrophages binding to MHC molecule, right? So this is MHC antigen complex. And this cell is called antigen-presenting cell. Now this macrophage gonna move to lymphoid tissue and is gonna activate helper T cells or CD4 cells. And what's shown over here with this arrow are interleukins. So when this activation happens, it starts releasing all these different chemicals, right, that affect the cells, that actually cause, you know, it stimulate macrophages, it stimulate T cells, and the most important, it's actually gonna stimulate both cytotoxic and B cells. And so that's helper T cells stimulate both cell-mediated and humoral immunity. Because if you go to this humoral immunity, if you go back to this humoral immunity, you don't see any helper T cells. So you're thinking like, okay, so here I have my antigens, they activate B cells start proliferating. They can do everything on its own. Yes, they can, but they do it very, very slowly. Actually, these cells, when they are selected, right, this one selected because it has receptor for antigen, it's gonna see it in the lymph node for a pretty long time. It's actually waiting signal from helper T cell. And when helper T cells send these signals, right, so that we have over here, then they start proliferating very rapidly. So helper T cells, very important in both cell-mediated immunity and humoral immunity, because helper T cells activate cytotoxic T cells and B cells. Okay, now organ transplants, there is 4 varieties. Autografts from like from one body side to another in the same person, right? Let's say when you have like skin graft, skin autograft, when they take a piece of your skin from one part of your body and you know, and transplant it to another part. Let's say if a person has like a severe burn. Isograft between identical twins. That's, we're not that lucky because not many of us have identical twins. Allografts between individuals who are not identical twins. So what we get rather than our own autografts would be allografts. And xenografts, grafts from animals, right? Or from another animal species. So when we get something from animals. So prevention of rejection, of course, when we have any graft, it will be rejection, some type of rejection, with exception of autograft. right? But even isograft sometimes can be rejected, but of course way less. So rejection depends on the similarity of the tissues. Patient is treated with immunosuppressive therapy, like immunosuppressant drugs, corticosteroids to suppress inflammation. So this is all suppress your immune system, so it doesn't allow your immune system to reject that tissue, foreign tissue. And many of those medications has severe side effects. Okay, so that was the organ transplants. Now, immunodeficiencies. Immunodeficiencies are congenital and acquired conditions that cause immune cells, phagocytes, or complement to behave abnormally. So every time when those cells behave abnormally, we call it immunodeficiencies. So here's one: severe combined immunodeficiency syndrome. This is genetic defect. Why we call it combined? Because it's a defect in both B and T cells. So it's combined because it, affects both cells. And it's fatal if untreated, and it's treated with bone marrow transplants. If it's deficit in the B cells and T cells, so that person does not have any specific immunity. It does have some first and second line of defense, non-specific or innate immunity, but no specific immunity. Hodgkin's disease is a cancer of B cells, and it leads to immunodeficiency by depressing lymph node, and this is where inside lymph node B cells divide, right? So if you have cancer of B cells, so the person has most problem with the humoral immunity. And of course, cancer, if it metastasizes, can affect other organs. AIDS, right? Very famous acquired immune deficiency syndrome caused by human immunodeficiency virus, HIV, transmitted via body fluids, blood, semen, vaginal secretion. And it's also can enter via body transfusion, blood contaminated needles, sexual intercourse, oral sex. And HIV virus, what it does, it destroys T helper cells, right? So you can see that if T helper cells are destroyed, it depresses mostly cell mediated, because the patient still would have humoral, right, immunity, but it will be very slow. Treatment with antiviral drugs and reverse transcriptase inhibitors. We're not going into detail, but because it's retrovirus, it doesn't have DNA, it has RNA. So first RNA need to be transcribed into DNA, and this is what inhibits this enzyme. So it doesn't allow RNA to be converted into DNA, or not converted, but transcribed into RNA. Also, protease inhibitors. This one destroys viral proteins and doesn't allow to assemble parts of the virus. Also some new fusion inhibitors that block HIV entry to helper T cells. So we have a huge progress in this now, right? So people, we cannot kill this HIV virus, but we really can suppress it so much that people can pretty much live a normal life now. But we cannot kill the virus. It's so, I would say, sad that with all our technology and everything, that we're trying to do, we still cannot kill viruses. As I say, how you can kill something that's not considered to be alive? That's what virus is, right? So we cannot kill HIV virus, but we can actually manage this condition pretty well now. Now, autoimmune diseases are diseases of your immune system. When your immune system loses ability to distinguish self from foreign, and it's synthesized T cells that destroy your body tissues, or it can be even antibodies that binds to your own cells. So examples include lots of diseases like multiple sclerosis, myasthenia gravis, Graves' disease, type 1 diabetes mellitus, systemic lupus erythematosus, glomerulonephritis, rheumatoid arthritis. But what it says, it actually can affect your nervous system, your muscles. Graves is your thyroid gland. Diabetes mellitus is your pancreas. It can affect your skin. It can affect your kidneys. It can affect your joints. So pretty much your immune system can affect different organs of your body, and based on what organ it affects, it has a different name. Like multiple sclerosis, that's a nervous system. Like Graves' disease, that's a thyroid, right? Glomerulonephritis, kidneys, and so on. Hypersensitivities, immune response to perceived otherwise harmless threat, causes tissue damage, and antibodies cause immediate and subacute hypersensitivities. So hypersensitivity is what we call allergy, really. So it can be acute or type 1 hypersensitivity, or this is what we call allergies. And it usually begins in seconds after a contact with allergen, and subacute, slow onset, 1 to 3 hours, but it has a long duration. And this is different antibodies involved, like here, immunoglobulin M and G. And the last one we cover here, anaphylactic shock. Anaphylactic shock is a systemic response to allergens that directly enters the blood. So it's one thing when we get this, allergen that enters our mucous system, right? Our skin. But when it enters our blood, that can cause huge and very dangerous reactions. That's what anaphylactic shock is. Now, basophils and mast cells, they when this happens, right, they start secrete lots of histamine. And what histamine does, histamine vasodilate your blood, and when blood vessels are dilated, you actually have a huge loss from bloodstream. So not enough blood flowing to your organs, especially to your brain, because you have like limited amount of blood, like 5 liters of blood, and it's inside your blood vessels. Now, when all those blood vessels became huge, you kind of don't have enough blood inside blood vessels. So very little blood delivered to your heart, to your brain, and that's what causes actually shock and death. And it's also constrict bronchioles. Like that was not enough to vasodilate everything, right, and reduce blood flow to the brain. It's also constrict bronchioles. And the treatment is epinephrine, because epinephrine is a vasoconstrictor and it will dilate bronchioles. Okay, I think that's it for this chapter. So it was chapter 21, immune system. And thank you for watching. I hope it was helpful.